So we're going to move on to the next session, which I've explained during the program overview session briefly about the Equip VentureLift grant that we offer. So in this session, we are providing a short lightning talks by the Equip VentureLift Awardees, who's gonna be speaking a little bit about their innovations and ventures. So they have actually just 3 minutes of the lightning talk, followed by a 2-minute of question and answer. So we'll begin. I know we're pressed by time. So with our first speaker, which is Mark Shinuda from Neuropeutics, Inc.
Every minute around the world, 2 people are being diagnosed with a neurodegenerative disease, something like Alzheimer's, Parkinson's, or ALS. And these patients are being told that there is very little that can be done for them. ALS is such a disease that is fatal, and 14 people receive their diagnosis each day, and they're being told that they only have 2 to 5 years to live. And over these years, they start losing their ability to talk, to walk, and then finally to breathe. The current treatments are only offering about a 6-month life extension, which we can all agree is highly inefficacious. One of the root drivers of the disease is a protein called TDP-43, which, as shown here in green, is in the center of neuron. But what happens in disease is that this protein leaves its normal location, forming these toxic clumps. And so 2 disease mechanisms are at play: the loss of function from the nucleus, but also the gain of function in the cytoplasm. And this leads to neuronal death. And so at Neuropeutics, we have developed a first-in-class patent-pending small molecule called JRMS-22, which not only clears these toxic clumps, but is actually also able to reverse the protein clumping, allowing the protein to re-enter the nucleus and resume its normal function again. Our molecule is highly efficacious, reducing disease pathology by about 30% in a humanized mouse model only after 2 weeks of treatment. Compared to other FDA-approved therapeutics, we are targeting the pathology at its root rather than secondary pathomechanisms that are arising from the initial presence of TDP-43, and as such, we expect to extend patients' lives by years. We are addressing the ALS patients in the USA as our initial beachhead market, following that by global ALS therapeutic market, then extending more broadly into other indications in neurodegenerative diseases. Our team has over 150 years of expertise in ALS research, diagnosis, clinical trials, and we are working diligently to advance to clinical trials in the next few years. Our biggest partnership has been with LifeArc, really supporting us with over $10 million in in-kind funding to really advance IND-enabling studies, followed by AbbVie, the Biotech Innovator Award earlier this year. We're currently finalizing our optimization and starting our IND-enabling studies by 2027, and then initiating clinical trials that combine phase 1b/2a by 2029. We are currently raising a pre-seed round that is led by TIAB that will really bring us to this IND acceptance by end of 2028. So with that, thank you for your attention, and I hope you join us on our journey to bring a new effective treatment for ALS patients. Thank you.
If anyone has a question, can you put your hand up? Maybe not? Okay, then we'll move to the next one. Thanks, Mark. So, you have a question?
There's someone here. Well, here.
What is IND enabling?
Oh, all right. Continue. Continue the question.
I was just wondering what IND enabling studies is.
So IND is the investigational new drug something. I can't remember the D, but it's just the first application that you submit to the FDA to initiate clinical trials.
Thank you.
Thank you.
Was there a question there? You're good. Okay, thank you, Mark. So then the next, The next person to present is Ethan Bidaki from Thermocular AI Corporation. And let me pull up the presentation. Thank you. Take it away.
Hello, everyone. I'm Ehsan, CEO and co-founder of Thermocular AI. University of Waterloo spin-out transforming how clinician can detect and manage dry eye disease. At Thermocular, we deliver a rapid, objective dry eye screening in under 30 seconds, something that has never been possible in routine care. In the next few slides, I will show you the problem we are solving, how Thermocular works, and why clinicians are eager to adopt it. Dry eye affects over 120 million people in North America, costing over 60 billion every year. More than half of the people are undiagnosed because the current tests are slow, subjective, and expensive. When dry eye left untreated, it can cause the system worse, and it can cause eye irritation, vision blurs, and in some severe cases, it can even damage the cornea. To validate this, we spoke with over 100 clinicians, and the message was clear. If this device could screen in 30 seconds, they would use it on every patient. And it's already in traction. We are in talk with 5 clinics and aiming to reach 10 by early 2026. We also tested the device in 78 patients to validate it clinically, and it shows that it can reliably measure ocular surface temperature and distinguish dry eye patients. Thermocular is a dual camera system, a visible and thermal camera that can capture tear film behavior in real time. Thermocular can do the test in non-invasively under 30 seconds and can easily fit into a routine eye exam. Thermocular is designed to fit into Class 2 FDA and Health Canada medical device requirements. Our workflow is straightforward. Our software analyzes the tear film cooling behavior, and after that create a clean objective report for clinicians. And clinicians can use it to make a decision about dry eye more confidently in seconds. We built a good traction from patent to prototype. We already spoke to over 100 clinicians, tested the device on 78 patients at the University of Waterloo, and we're preparing documents for FDA and Health Canada submission to submit it in 2026 and aiming to launch the product in 2028. I want also to thank BioHubNet for the support. The support helps us to complete clinical validation, to advance our regulatory planning and device optimization, and it also helps to bridge our research prototype toward market-ready product. We start with 30-second screening device, and we are expanding to full ocular surface diagnostics tomorrow. Our vision is clear: to make advanced ocular surface imaging accessible in every clinic worldwide. Thank you.
Is there a question from an audience? Okay, thank you for the presentation. The next presenter is Narges Al-Arabi from VentureLift, Inc. I just need to get to... There you go.
Good afternoon. My name is Narges. I'm a postdoctoral fellow at Toronto Metropolitan University and also founder of VentureLift. Infertility is not just a statistics for me. It's a personal issue. I have watched my friend going through many years and also a lot of spending on the fertility treatment, and the only reason for that was the male infertility issue. Male infertility issue is unfortunately on rise. 1 in 12 couples in North America are challenged by male infertility. The very first and the most recommended treatment is in vitro fertilization or IVF, And IVF, I think many of you might have heard of, has a success rate of 35%, is aggressive, expensive, costs $15,000 per round, and have the chance of multiple births. The second treatment that is inefficient, unfortunately, for the cases of male infertility is called as intrauterine insemination, or IUI. IUI involves separating the healthy sperms from the non-healthy ones and inserting it into the uterus. Unfortunately, for the cases of male infertility patients, the process of sperm prep is very inefficient and causes more sperms to die during the process. This is the very reason that the experts are pushing couples to do IVF. Here at VentureLift, we are targeting this issue with, we would like to make the IUI more accessible and also efficient. We have changed this dilemma by suggesting a device called as fertility tab. This device is mimicking the natural sperm selection that is done in the female partner's body, and we just do it in a microfluidic, small device. It's a US patent pending technology, and it separates 20% more healthy sperms than conventional methods that is currently done in the clinics, and it does it in 33% faster time. And we expect that by using this technology, the success rate of IUI can be boosted and can be near to that of IVF. And I can say that using this technology can save only Canada market up to $60 million per year because of the unnecessary IVF that is recommended for the male infertility cases. We are very thankful to BioHubNet for giving us the VentureLift grant. We are validating our beta prototype using this grant. We have done some testing with the animal samples. We are expecting to do the clinical validation of our MVP by next year, and also getting ready for doing the FDA submission by early 2027. And on our path to the market, we are looking for getting funding through the non-dilutive sources and also investment. Thank you very much for your attention.
Is there any question for Nargis? No questions? Okay, thank you so much.
Thank you.
So then we'll move on to the next speaker, Jonathan Labriola. He was here, sitting here, giving the presentation, but to his the actual company, you get to learn more about it. So take it away.
Yeah, thank you. I'm happy to talk about where I am now and what we're doing at Synakis and what makes it so exciting. So retinal detachment is a sight-threatening injury that requires immediate surgical intervention in order to preserve the patient's vision. Right now, the procedure and the materials, devices used are efficacious. What happens is the surgeon needs to go into the eye cavity, remove the native vitreous from there. It's a jelly-like substance, so they can access and repair the retina. Once that's done, they deliver a material into the cavity to hold the retina in place while it heals. And as I mentioned, these materials work. They hold the retina in place, and the patient's vision, the retina heals, and the vision is often recovered. However, the materials themselves impose extreme discomfort on the patient while they are healing. So, for example, they don't have the transparency and refractive index of the native vitreous. And so while this material is present, which can be weeks to months, the patient can't see, they have blurred vision. Because of the physical properties of these materials, in order to have them push against the retina, they need to posture, sometimes for up to 16 hours a day, perhaps face down or tilting their head one way, so that the material can work, hold the retina in place, and allow it to heal. So you can imagine this is extremely cumbersome. The patient can't travel or work or engage in really any activities aside from head posturing. And finally, in the case of some materials, they're not naturally resorbed, and so another surgery is required to remove the material once the retina is healed. And this is an injury that's quite prevalent. About 3 in 100 people will experience this in their lifetime. It happens as patients age. So to solve this problem, Sinakis invented SNK125. It's made of hyaluronan, which is the material that is also present in the native vitreous, or that comprises the native vitreous. And so because of that, it has the same properties, namely its transparency and refractive index, so the patient can see while their retina heals. We say it's biomimetic, and what that means is it has the stiffness and other properties that allow for it to hold the retina in pace without the need for any of this posturing. And so the patient can go back to normal life once they have the surgery, engage in all of the activities that one would want to engage in. And finally, it's biodegradable and resorbable, so it naturally degrades and is resorbed by the body over time. So no secondary surgery is required to remove it once the retina is healed. And this translates to a rapid and comfortable recovery for these patients experiencing retinal detachment. So this is our preclinical development plan. We're right at the phase where we've mostly frozen our CMC processes, and we're getting ready to scale up and do our, because this is a medical device, it's called an IDE enabling studies, and that will allow us to obtain regulatory approval to do a first-in-human safety study in Canada, and then ultimately we want to go to the US FDA to commercialize and market the product there. We already heard today about the Valley of Death, this space between where government funds for academia run out, but you're not quite at the phase where you're going to get funds from VCs, which is usually when you're at the clinic. And so we're currently actively fundraising to get us through that phase. We're looking to raise about $5 million in capital for that purpose. With the help of BioHubNet through their Venture Equip program, we've started on this process and are going to continue that as we raise. And so with that, I want to thank BioHubNet for helping us turn this into this.
There's our question, Gilbert.
So can this, you talk about it being used after retinal detachment, but sometimes before retinal detachment, there's a vitreous detachment, right? So can this be used then as a protective measure to prevent retinal detachment? And what are the economics and risks around that, right? So earlier injection of hyaluronan in order to prevent, to reheal or to refill the vitreous, right? And to prevent vitreous, retinal detachment.
Yeah, that's a great question. So as Gilbert mentioned, the native vitreous is stuck to the retina and it degrades over time, and as it becomes loose, it can actually be the cause of the problem, pulling the retina off. That's a really interesting point. You injected and fill that space and kind of prevent that. I think it could work. It's a good idea. It would have to be the sort of thing where you keep injecting it because our polymer degrades a bit more quickly than the native vitreous. But it's certainly something we could think about. It has some of the properties. It's injected as a liquid, so it can fill those spaces and potentially act as a preventative measure. Yeah.
Thank you, John. All right, so next speaker is Olga Klushna from Oculum.
Well, maybe I can start while figuring out the presentation. But looks like Jonathan made a great introduction for retina. I'm going to continue into the retina space as well. Hopefully, because like my first line really need that picture. It was like a punch to get your attention there. No worries. Yeah, please do.
These things happen. I think it's because it's PDF.
I have a PPT as well.
Just dragging works. Nope. Oh, there we go. Look at that. Oh, it's coming up. Oh, almost there. Awesome.
Yay. So the image in front of you is what the surgeon sees through the microscope during a surgery. Here, they're manipulating transparent and very delicate tissues of the retina with a forceps. Can you see exactly where the membrane have been peeled? Probably not, because it is transparent, but the surgeons are asked to see the invisible every single day. So how do they overcome this problem right now? So since it, sorry, I'm gonna be, here we go. So currently, a chemical stain is injected into the eye, and that's what surgeons use for guidance. Sounds simple, but last year I observed hundreds of surgeries, and that process is far from simple. It takes about 10% of the surgery to inject and prepare the dye. Also, the results are highly skill dependent and very patchy, but reported about 40% of the surgeons. After this is done, the dye have to be flushed out to reduce, but not eliminate the side effects, such as retina toxicity and steroid-induced glaucoma. To put it bluntly, any chemical stain will negatively affect the recovery. So Oculum, sorry about that. Trying to get to the final one. So could you scroll thing? So Oculum is the next one. Oh, awesome, thank you. So Oculum is taking this challenge into 21st century by introducing a virtual stain, which is AI-enabled visualization system, which is in real time can guide the surgeons through the peel of the transparent tissues. Our technology will give a consistent and reliable visualization, and it is a modular add-on to an existing surgical microscope. That means we do not interrupt the clinical flow. We would like to say thank you to the BioHubNet because they will help us to continue and finish our second clinical study, which we're currently doing at Sunnybrook Hospital, where we will validate further our software, and as well, BioHubNet will be very instrumental into finishing our first fully functioning prototype. Thank you so much.
Can we have an extra applause for Olga for overcoming that technical difficulty? But thank you so much. Is there a question from an audience? Oh, there's one.
So could you just clarify, are people already using this in human patients? Is this already being used in human patients?
Not yet. Oh, sorry, not yet. It's not a clinical trial, it's a clinical study.
A clinical study, thank you.
Yeah, so basically, we currently getting most of the information. We have about 9 patients like data acquisition, but that's what we like using to actually train our algorithm and so on.
Cool, thank you.
Thank you, Olga. You did great. So then the next speaker is Stephanie Burek-Eggers from Spark Inc. And we have your slides.
Hi, everyone. I'm going to share a bit of a story with you. Not long after my undergrad, my parents took me to an athletic banquet. And my father was athletic himself, was particularly proud. And that evening I came home to my proud athletic father, only he was having a stroke. And 5 days after, he died. And there were no signs, no symptoms, no way of knowing it all. And unfortunately, I learned very quickly after that experience that I wasn't alone in that experience. And that when we look at cases like stroke and cardiovascular disease, compared to COVID-19, for example, at the peak of the pandemic, cases are 4 times more common with cardiovascular disease, and deaths are 7 times more common. And if we don't intervene now, then by the year 2030, those rates are going to rise to 23.4 million per year, cardiovascular deaths per year. But really, the most interesting part is that many of these diseases are preventable, and some are even reversible if detected early enough. But this early detection is not easy. And our company's solution is a handheld device. It screens for risk of cardiovascular disease using the saliva sample placed directly on the device. Results are offered rapidly in under 6 minutes and without the need for technical training or traditional laboratory. The device captures a select set of protein biomarkers directly on the device and indicates a user's level of risk as low, moderate, or high on the readout screen. And this type of screening can prevent up to 3 million cases per year, 75,000 deaths per year, and that's in the United States alone. And we've been able to support our growth through our partnerships, including H2I, who's here today with illustrated through our grants, our prizes, our awards, including being named the #1 early stage company by U of T Entrepreneurship, which altogether has allowed us to remain 100% non-dilutive with over $150,000 in funding. And we're translating all of that support continuously into action. And so, for example, we're now with SpinUp, the U of T's first wet lab incubator, where we've launched our lab there, hired our first full-time lead engineer, identified our biomarkers of interest, and validated our prototype with human saliva, and then also filed our provisional patent with funding secured through IPON. And if you're an entrepreneur and you haven't used IPON yet, I urge you to go check them out. And then with the VentureLift Award is continuing this support, and we're excited to be able to use this funding towards, we've been able to access regulatory compliance or regulatory experts towards regulatory compliance. We're initiating our MVP manufacturing, and then also accessing essential equipment, materials towards our trials that are getting us to our clinical pilot, and leveraging it towards more funding. All led by this team of experts in engineering, in cardiology, in salivary diagnostics that have come to this company with over 300 clinical trials in their pockets already, experience with patents, with licenses, and health tech ventures. But most importantly, all with this shared understanding that we can't begin to conquer a disease until we can detect it quickly, early, and in every corner of the world. Thank you.
Thank you, Stephanie. Is there any question in the room? Oh, I see.
Thank you. So the question was, what's the marker we're using to detect this heart disease? We're actually have several biomarkers that we're working with, and some of which are specific to diseases, specific diseases like heart failure. Others are non-specific to cardiovascular disease, are clinically relevant to cardiovascular disease risk, but not necessarily specific to cardiovascular disease itself. So both categories we're working with, and there's 2 leading ones that we've done already with our device that it's detecting and stratifying. Yeah.
What's the efficiency for this? Like, how efficient is it, like, to detect the heart disease with this device?
So while adding this to a traditional risk screening profile, you can increase your area under the curve of 0.5 to 0.7 right now with the 2 that we're leading with in our lab at the moment.
So is the goal to sell it to consumers or to doctors, or how's that going to work?
Yeah, the team we've been working with the regulatory specialists as well as the team with lab to market launch. And together, the go-to-market strategy right now is to start under the FDA wellness policy in order to get and penetrate market as quickly as possible and start collecting our data with a direct-to-consumer type product. And then in the background, pursuing this regulatory compliance and getting it into the hands of clinicians.
We'll take one last question.
So I saw that you have a cardiologist on your team, but I'm curious about, since you're selling it toward consumers, if a positive result on a device like this is sufficient to get a doctor to pursue further testing or treatment options if this is, I guess, not necessarily diagnostic, but sufficient for encouraging further steps?
Yeah, so the direct-to-consumer version of this, the wellness policy version of this that would be covered under FDA wellness policy, that is with biomarkers that are already clinically validated and stratified across this low, moderate, and high risk. So clinically transferable to exactly what's already being used by a clinician. So unlike something like you would say, perhaps, let's say, with 23andMe, when people were coming in with these results that weren't necessarily aligned with what a clinician practices with every day, what would be going to market first would be those that are transferable directly to what they use in their office already.
Thank you, Stephanie.
Thank you.
So then the last presenter to wrap up our spotlight on VentureLift Awardees session is Avantika Vaida from VRIT Inc. So she is actually presenting on behalf of Sushant Singh. He was one of the round 2 VentureLift recipient, but he couldn't join us in person today. So I'm just gonna transition to your slides quickly. And she stepped up on a very last minute. I think she had a 1 day to prepare it, so we'll give it another round of applause.
All right, good afternoon, everyone. My name is Avantika, and I'm an associate at VRIT, where we're developing next-generation skin bioprinting technology for both defense and civilian healthcare. So at VRIT, our vision is to transform the treatment of skin injuries from skin graft replacement to on-demand skin regeneration, making it possible to heal skin directly on the wound site without needing to harvest donor skin from anywhere else on the body. So the current standards in burn and trauma reconstruction remain autologous skin autografting, which is a technique where you take healthy skin from the body and apply it to the wound site. And this technique is centuries old at this point and is still widely used. However, these grafts can fail in up to 30% of cases, cause significant donor site pain, and often result in visible scarring. And as the wound area increases, so do recovery times, costs, and complications. Since then, several dermal substitutes and skin composite technologies have emerged. However, these unfortunately still require multiple surgeries and fail to replicate full living tissue in a timely manner. So what's needed ultimately is a technology that can restore the skin barrier rapidly, safely, and effectively at the point of the wound site, whether that's at a specialized burn center, a rural hospital, or even a deployed field environment. That's where VRIT's dual technology platform comes in. Our first product is Insight, which is a handheld bioprinter that allows clinicians to conformally deposit biomaterials directly onto the wound site, creating smooth, even coverage without stitching or grafting. It's designed to be portable, intuitive, and compatible with current surgical workflows. Our second product, Shield, is an off-the-shelf cell therapy, a ready-to-use lab-grown skin material that's available on demand and can be directly deposited onto the wound site. Together, these technologies enable a shift from replacement to regeneration, with the potential to reduce infection, shorten hospital stays, and eliminate the need for donor skin entirely. By combining portability, precision, and biological repair, VRIT's platform bridges the gap between advanced hospital care and austere environment care, where rapid wound coverage and recovery can make the difference between healing and severe complications. We're supported by a strong network of institutional and industrial collaborators who recognize the value of this technology in diverse settings. And our advisory board brings together global leaders in burn surgery, biomaterials, and medtech commercialization with scientific advisors whose previous companies have been valued at over $3.5 billion. Ultimately, our team is working together to translate years of research into real-world impacts. Through the BioHealthNet VentureLift Award, we're using these funds to hopefully conduct a health technology assessment so that we can get evidence-based insights to further inform our technological and preclinical validation. Ultimately, at VRIT, our vision is bold but simple: to make on-demand skin regeneration the new standard of care. And we believe we can redefine how the world heals skin with effective technology, partnerships, and support. Thank you.
Is there any question in the room? I see John. Oh, sorry. Okay, and then after.
Are there any concerns about immunological rejection about this material?
Yeah, so I've been given strict instructions to be careful about the cell technology, but that's definitely a priority in our development cascade to promote immunomodulation and obviously create a therapy that can safely integrate into the physiological system.
That was a great talk. Do you know how this will be regulated?
Yeah, so currently for the hardware side of the printer, we've identified a 510K pathway. So there is a potential predicate available. As far as the cell therapy goes, that would be the highest drug classification. So all 3 phases of trials would have to be gone through.
Thank you.
Thanks.
So I just want to thank all our VentureLift awardees for fantastic presentations. I know that 3 minutes is definitely too short to share all the details of your amazing innovation and technologies that you're building, but it's wonderful to see the progress that you're making, and we are proud to be part of that, your entrepreneurial journey. So thanks again for all the VentureLift awardees.